Conexão estabelecida. Estamos ao vivo. Boa apresentação, Shabir.
Obrigado, Silva. Só esperar aqui aparecer no YouTube, a gente já inicia. Fico acompanhando no YouTube, eu gosto de esperar aparecer no celular para... Okay, we are online. So, good morning to everyone. Today we have the presentation of the data seminar from Shabir Ahmad that performed his master degree on our program under my supervision. For those who are present today in our seminar and are not familiarized with our program, this seminar is presents the preliminary data that the student has for his master or PhD. So we will have now an image of the main results Shabir has been obtaining during his master. And after the presentation, we have time for some questions from the audience. So Shabir, I will turn off my camera and you please may start your presentation.
Okay, thank you. Bismillahirrahmanirrahim. My name is Shabir Ahmad and I'm working in the lab of structural bioinformatics under the supervision of Professor Dr. Guarli. And today I'm going to present my data seminar about my master, and that is about the genomic, transcriptomic, and structural wide analysis of one of the tick, that is Rhizophilus microplus, for the prediction of some stable anti-tick vaccine, and some drug profiling for one of the tick-borne pathogen, that is Rickettsia prowazekii. So first, I would like to introduce about the backgrounds of the ticks. So ticks are the obligate blood-feeding parasites that cause a lot of disease and transmitting pathogens to humans and animals, and cause a lot of public health and agriculture sector loss, create a lot of economic loss to the all over the world, especially to the agriculture countries. If we can say about the Brazil, so they are causing $3.2 million annually in Brazil. Basically, ticks cause parasitism by two means of way. One is the direct way, and another one is the indirect way. So the direct way is basically they cause skin injuries, anemia, blood loss, and the reduction of the milk productions. While indirectly, they transmit a lot of tick-borne pathogens such as Babesia species, Theileria species, and Anaplasma marginale. Scientists are currently using chemical acaricides, the current strategy to control these ticks and tick-borne pathogens. But we have a lot of populations tick resistance across the world, and some of the studies have been confirmed these resistance. I have just shown in the map about some of the acaricidal resistance studies in the Brazil that is shown in the figure. Alternative way is to control these ticks and tick-borne pathogen is to is the process of immunization. So we have a lot of antigens identified before, in which BM86 is one of the antigen-based anti-tick vaccines that cause 80 to 89% tick controls. And we have a lot of more unique antigens that is more than 40 and have comparable efficacy to BM86. A lot of them have shown comparable efficacy to BM86. But we need a vaccine that can control multiple ticks at a time. So we need to target a new targets for anti-tick vaccines. So previous studies have been published using this approach, which I'm going to discuss about other organisms and ticks too. Another aspect is about the Rickettsia prowazekii, that is the drug reprofiling Murray enzyme. So Murray enzyme is a target for the broad spectrum antibiotics, and there some species are resistant to this drug fosfomycin, while some are inhibited by these drugs. So the species that are resistant to these fosfomycins, which is a drug, is a Mycobacterium tuberculosis and Chlamydia trachomatis. while Streptococcus pneumonia was inhibited by these drugs, which is fosfomycin. So the first question here arises why we choose this enzyme. So this enzyme is basically predicted in our previous published work as a drug target, and this enzyme is found in most of the Rickettsia species. So the objective of my work is basically to characterize some potential stable injected vaccine against the Rhizophilus microplus and drug repurposing against one of the gene of the Rickettsia prowazaki, that is Murray gene. And my specific objective is to analyze the genome and transcriptomic of the Rhizophilus microplus to predict essential genes that can be used as injected vaccines. So for that, we need to find some non-homologous proteins. So we need the non-homologous proteins for the Rhizophilus microplus, and after we need some of the subcellular localization of these proteins. So we predict subcellular localization of these proteins, and the final specific objective is to study the inhibitory effect of one of the drug, that is fosfomycin, against the gene's Murray enzyme. So the methodology for the ticks, that is Rhizophilus microplus, so we downloaded the transcriptomic data of the Rhizophilus microplus in FASTA format, that is almost contained 10,932 proteins. And we uploaded this data set to a software that is called CD head analysis, and to get non-paralogous proteins and remove the paralogous protein. They have some threshold values, that is 40% to up to 1%, up to 100%. So mostly people use 40%, but we use the strengthened thresholds to get to narrow the data, because we have a lot of proteins. So we use the 60% threshold here, and after that data, when we get the non-paralogous proteins, we uploaded that proteins to BLASTP, standalone BLASTP, to get non-homologous protein, and we use the Bostaurus as a host here for Rhizophilus microplus. So we BLASTP our transcriptomic proteins against the Rhizophilus, against the Bostaurus, which is a bovine. And we get only non-homologous protein that is only present in the Rhizophilus microplus. And after we uploaded this proteins to the database of essential genes, which is a database contain a lot of essential genes from different bacteria, from different prokaryotes, from different eukaryotes. And when we get the essential genes, we uploaded that to another software that is called Cascade pathway analysis, to analyze the pathways of these proteins, that in which pathways these proteins are present, these enzymes are present, these genes are present. And after, when we get some of the pathways specific proteins that is present in pathways, so we uploaded that to another server, that is QuakeGo, which is to analyze the gene ontology of these enzymes. So we uploaded that to QuakeGo analysis, and we use a batch script for this software to upload all the genes, all the enzymes, all the proteins, to get only those proteins which have the function in the database and have a cell membrane localization. And we use the same proteins to another software, subcellular localization, which is Cello, to get only those proteins that have the cell membrane qualities, because from the literature it is said that the cell membrane proteins can be used as a vaccine candidate. And after, when we get subcellular localizations, we choose only the membrane proteins, and we then uploaded it to another software that is called Vasagen version 2. To find the antigenicity of these proteins, that how some of these proteins are antigenic, some are non-antigenic. So we need to choose only those proteins which are antigenic, and we use the thresholds here 0.6 again to have high thresholds. So we get only the best antigenic candidates. And after we model some of the antigenic protein using a server from DeepMind, that is AlphaFold, to predict the three-dimensional structures of these antigenic protein. And about the flowchart, about the drug target, that is the fosfomycin and the Murray enzymes. So we downloaded the Murray enzymes FASTA sequence from the NCBI, and use as a BLASTP against the PDB database to get some best template. So we have the template 1UA, And we model these proteins using the model 9.2 versions. After we adopt these proteins to the drugs using the Actor web server, and we get the best docking pose. And after we do the molecular dynamic simulation of these complex to check the stability of these complex. So we run this molecular dynamic simulation in triplicates. We use the Gromos version, and the pore speed was Gromos S96 version of 54S7. Sorry. We use the ligand topology was built with the ATB, that is automatic topology builder. And the results from the first of that is the Tetracephalus microplus, that is the transcriptomic data analysis. So here is the flow chart, how we get the results. So the transcriptomic data contain 10,932 proteins. We uploaded that proteins to the CDH server, and we get only 7,536 proteins. That is non-paralogous. And these proteins we uploaded again to BLAST-P, and we get only 4,613 proteins that are non-homologous. And after we uploaded that to the database of essential genes to get only those genes which are essentials for the survival and like have some roles in the pathogen transmission are required for the survival. And after we use the KIT-Cas pathway to only get those proteins which are pathway specific, so we get only 1,362 proteins that are pathway specific and have roles in most of the pathways and present in the pathways. And after we do quick go analysis to get the subcellular localization in only those proteins which have the gene ontology, so we get only 785 proteins. And we uploaded again this proteins to the another subcellular localization server, that is Celo. So we get only 80 protein that have like the BLAST goal results and the subcellular localization we combine to get only those proteins which have the subcellular localization membrane proteins and are present in both servers. So we have the strengthened data. And after we uploaded this 80 cell membrane proteins to check the antigenicity of this protein using Vesigens, so we get only 37 protein as a final target up to now, which can be used as which can be tested as an anti-tick vaccine. The subcellular localization, we have 52 cytoplasmic protein and 80 proteins are membrane proteins. So we are only interested in the membrane proteins, that is 80 proteins. The antigenic proteins, we get only, as I show in the last figure, we have only 37 antigenic proteins. And basically this approach for subcellular localization in combination with Vesigens results in the inclusion of BM86. As shown in the literature that BM86 proteins are membrane bounded and they confer protective immunity and have the antigenicity, but it is not necessary that all 37 protein will have confer the protective immunity. So we need to test that as experiment to check the protective immunity of these 37 proteins. These are some of the antigenic protein in the figure. We have the in the table. These are 37 proteins which we predicted as antigenic proteins. And we are going to further analyze these proteins as a 3D structures and the B cell, and to find the B cell epitopes in these antigenic proteins. These are the 3D structures of some of the antigenic proteins we predicted up to now. Some of them have transmembrane, and now we are coming to the results from the Murray enzymes, that is which we dock to the postmomycin drugs. So up to now we have got only the results from the substrate, that is the UD1. We dug the substrate to the PDB, to the template. Up to now we have dug that complex, and in future plan we are going to check the stability of the postmomycin 2 to dog to the proteins to check the stability of the protein. After we did the molecular dynamic simulation of the complex from the substrate, so we this figure shows the average RMSD of the triplicate simulation, that is 100 nanoseconds, and we are extending it to 1 micro. So basically the RMSD is quite stable. You can see from the figure and the standard deviation of the figure. And after this figure, we superimpose the 10 nanosecond frames from the molecular dynamic simulation trajectories to check the movement of the loops and the movement of the trajectory. So we get quite stable RMSD from the figure, and after we did the differential RMSF from the trajectory simulation, as you can see from the figure, it slightly shows the flexibility, increase the flexibility, and here it's again decrease flexibility. So the ligand is quite stable in the dog complex, and it shows that we have a little loop mobility, but overall the structure is quite stable. So we are now planning to do some experimental work of these drugs using against the Rickettsia-Prowazaki, and we are going to using molecular dynamic simulation to dog the murine, to dog the phosphomycins, and do some molecular dynamic simulation to check the stability of the drug target on the murine enzymes, to check the stability. Now we are moving to the conclusion. So we predict some essential non-homologous proteins, and after that we find some cell membrane proteins with subcellular localizations. We also found some antigenic proteins, that is 37 proteins, that can be tested as antigenic vaccines to check the protective immunity against the Rhizosphere microplus. And about the murine enzymes, we did the molecular dynamic simulation of the sample and the dog complex to show, and it shows quite stable structures. And the folding is basically maintained in the simulation, and see a little loop mobility surround the active site, but the overall structure is quite stable. So we are planning to do some experimental works with these drugs against these murine enzymes. My future perspective is, as we have already modeled some of the three-dimensional structure of these antigenic proteins, but some we are going to model, and to identify the B-cell epitopes in these antigenic proteins, to show that how they have the B-cell epitopes in the structures, in the 3D structures, and how they can interact and confer protective immunity. And to dog the phosphomycin drug into the murine enzymes, up to now we have only dogged the substrate into the murine enzymes, but now we are going to dog the phosphomycins and do the molecular dynamic simulations again. To perform the molecular dynamic simulations of these drug complex against with the drug target, and to test experimentally the drug phosphomycin against Rickettsia-Prowazaki, and we are planning that now to test that experimentally in these drugs against the Rickettsia-Prowazaki. Thank you so much. This is our lab member, our principal investigator, Professor Dr. Guwarli, some of the postgraduate students and the graduate students in our lab. We acknowledge CNPQ here in campus, and is Guamong from that's all, and thank you so much. If you have any questions, you can write. Thank you so much.
Thank you, Shabir. Thank you a lot. Well, we arrived at more than 30 people in the presence, so I think you had a very nice audience. And I think it's important to give some perspective on Shabir's work. And he arrived on Porto Alegre right in the beginning of the pandemics. And I think when he put his feet on the university was the beginning of the quarantine. So most of his work was made during the quarantine, and staying in a foreigner country is already a challenging thing. So isolated is even more challenged. So I think it's important to recognize the amount of results you obtain in quite adverse conditions, to say the least. Shabir, let me do two questions about your work. When you show the image with the prediction of the 3D structures, can you put the slide for us to see? Yes. The next one. Yes. Most of these folds are alpha bundles. And you have in one case the presence of a domain with beta sheets, but in all other situations you have alpha bundles. Are all of these bundles transmembrane regions, Or if not, can we make some inference about why we are seeing so many alpha structures on these predictions, on these proteins, and not beta sheets?
Yes, I checked that with some of the transmembrane softwares, and they have transmembrane. Some have 4, some have 5, some have 2, some have more than 8 transmembrane. So I'm planning to organize it in the membrane to show the transmembrane and the structures, but I'm planning.
Okay.
Yes.
Okay.
Yeah.
So most of these proteins are transmembrane, and we are just seeing the transmembrane domain.
Yes, transmembranes.
Okay. And when you show a second question, when you show the RMSF, do you have a hypothesis about why this ligand interferes in the protein flexibility? Why does it increases the flexibility around, I think, residual 100 something, and reduce the flexibility in residual 150? You are muted, Shabir.
We have the active regions, active site region in the 114 to 121 in between. So that loop is moving in the simulation sometimes in the trajectory. So it's like the RMSD is increasing a little and then decreasing by the ligand. Yes, so the flexibility is a little in the loops, and because the loops is the active regions, and the ligand is docking at that place in the loop.
Okay, so it's a conformational modulation of the loop surrounding the active site.
Yes.
Okay, excellent. Shabir, you have a lot of congratulations in the audience. I'm glad to see some former students of us. You have a question from Professor Carlos Termione. Are these epitopes you identify located within the transmembrane regions, or they are outside the transmembrane regions, like in loops or in cytoplasmatic domains?
It's a good question. Thank you so much. But we are planning to find the epitopes in these regions. So it's a great question for me that I will find that these epitopes are in transmembrane or not, outside or inside, because we haven't did the epitopes still. We are going to plan the epitopes predictions now.
Okay, you will locate the epitopes.
Yes, the epitopes in the 3D model. Yes.
Okay.
Yes. No.
Okay. Yeah. Until now, you are just modeling and validating the models and taking care about the quality of these models.
Yes. I'm doing to have a good quality models. Yes, because they have very low similarity with the template. Like these types of protein don't have much templates. Like some have 15%, some have 20%. So AlphaFold is the best option so that we can model them in up to some accuracy.
Okay, that's important to mention. And some of these proteins are quite challenging to model with and AlphaFold, and you can end with poor quality models. So we need to filter that before doing any analysis. That's good to mention.
Thank you so much.
Okay. Well, I again thank for the audience for the presence to recognize Shabir's works. Shabir, thank you a lot. It was a great presentation. I think the data is quite mature, and it's clear for us to see the evolution of your results during this period in Brazil. I hope it has been a positive experience. Thank you a lot. Thank you, everyone, and have a good day.
Thank you so much, everyone. Thank you, Hugo. Have a good day.